please, fill those out. Okay, so what I want to do today is finish off on how herbicides are selective. And what you're going to see in the exam is situations where, you apply a pendimethalin or a dual, and the annuals are gone, but you've got these broadleaf weeds still there. And we talked about what are some of the ways that these herbicides work that make them selective. And we had gone through a bunch of examples. You can take advantage of plant height differences. Remember the sponge wick applications, rope wick applications. We had talked about differential penetration of the herbicide depending on the cuticle and trichomes and so forth. And we went through all of those. But really what we ended up, the last kind of slide we talked about is this, that really the major, reason why we have selectivity is that tolerant plants or plants that are not negatively affected by the herbicide are able to metabolize or detoxify the herbicide into components that are non-toxic. And the susceptible species obviously can't do that for whatever reason, whether it's selection or not, and they basically are killed. Okay? So it's really the alteration of the herbicide. It's not just taking it like linear on carrots, take it and put it in the vacuole and store it away as soon as it gets in. These guys come in and right away, there's detoxification going on in some of the resistant or at least tolerant species. And the example I'll cover is atrazine. Why is corn not affected by atrazine? Well, soybean is, and in fact some grasses are. And I'll show you that there are 3 pathways that corn uses to metabolize, break down the atrazine. So this is the important thing. All of these herbicides are degraded by enzymes, and that's why enzymes, proteins are so important. They're not there, you're going to have trouble. This is a little busy, but I just wanted to show you there are a number of chemical processes that occur that plants are capable of carrying out to detoxify or metabolize herbicides. And I'm just giving you examples. I'm not going to ask you to, again, regurgitate this, give me the structures, but I just want to show you some terminology that is used. And so things like, for example, 2,4-D, how do grasses, in your lawn, your fescues, your Kentucky bluegrass, why is it not affected by 2,4-D? Well, one of the ways that the grasses, what they carry out is called aromatic hydroxylation. Aromatic ring structure, hydroxyl OH group. So here in red, I'm circling what those plants are capable of doing. They basically they replace the chlorine here, with an OH group, hydroxyl group. And then this continues, and then they conjugate it, they add a glucose to it. And that product now, the 2,4-D, is no longer, herbicidally active. And then they're not showing it here, but this, conjugation will then, the plants break that down into smaller components, little amino acids. Some of them are used, some of them are excreted. But the chemistry is completely changed. The broadleaf weeds, the dandelions, plantains, they cannot do that. The 2,4-D goes straight, And what is 2,4-D? What is the mode of action of 2,4-D? Dicamba. It's an auxin mimic, so you'll see the twisting and bending. You don't see that in the grasses. Because they're capable of doing this. Now the question is, why are they capable and the broadleaves are not? That's a good question. It's just differential metabolism. When you get resistance development, which is going to be the next section I'm going to cover, you're going to see that some plants, for whatever reason, some broadleaf weeds, have gone through a mutation. There's been, an error in the DNA replication, and for whatever reason, they are capable of doing these processes themselves. And when you apply selective pressure, you use the same herbicide over and over again. That one individual that's a mutant, that's able to resist the herbicide or tolerate it, now becomes, instead of being 1%, 0.1% of the population, 10%, 20, 30, till pretty well all your population is of resistance. But that's one, of these, and you see it in some of the other herbicides. Again, I just wanted to give you a sense that it's not just something that we do know what happens, what the byproducts are. And the alkylation, an alkyl group is the CH, CnH2+1, you know, so what you're seeing here is basically in trifluran or treflan in resistant species, okay, what they're able to do is they basically remove this alkyl group and add a hydrogen to it, and that renders, okay, this product, okay, basically non-herbicidal, okay? That's why you can use it. Susceptible species can't, okay? So you've seen the treflan basically, okay, what happens to the corn. That's what happens, okay, but the soybeans are capable of carrying this process. So an alkyl group, this is an alkyl group right here, okay, the CH2, okay. A methyl group, CH3, the methyl group right here. This is monuron, sulfonylurea that you don't have to know, but just wanted to show you what it does. It basically, this resistant species or tolerant species remove they demethyl. They remove the CH3, the methyl group, and add a hydrogen to it. And those simple reactions, okay, allow basically the plant not to be affected. And then this, typically this is broken down to further, but I just want to show what that initial process is. Obviously, susceptible species cannot carry this out, and of course, monuron being a sulfonylurea is going to be affecting amino acids production, branched amino acids. So it inhibits acetolactase synthase, the enzyme, okay? And deamination, the NH2, okay, that's another sulfoxidation, metribuzin. Susceptible species cannot carry this process out, okay? You'll notice here what's happening, okay? There's conjugation. S-glutathione is an important, you know, it's like glucose, important conjugate, and when you have this, basically, it basically blocks the herbicidal activity, okay? So this is just, again, I'm not going to ask you, give me an example of deamination or demethylation, but just understand that it's not, you know, unknown. We do know what's happening in most cases, okay? So let me give you a concrete example, because this might be, it's a little abstract for some of you, not all. And so I want to give you the example, specific example of atrazine, okay? Remember, that's one of your, okay, the key herbicide, probably most widely used corn herbicide in the world, and definitely in the U.S., okay? Top 10, top 5, okay? And here's some work that was done showing the amount of atrazine that was metabolized to non-toxic forms 6 hours after treatment. Now, I just want to show you what happens. In 6 hours, corn can detoxify. 96% of the atrazine is detoxified, or it's broken down into non-herbicidal components. But look at some of these other grasses. That's why it does have some activity on grass. Of course, soybean gets hammered. That's why we have residual issues. We could have. But fall panicum, it's only 44%. Giant foxtail and oats, 2%. That's why atrazine, you wouldn't put it in oats. Again, grasses, monocots. You don't even have to look at broadleaf weeds. Okay? So what is it about corn that allows it to break down atrazine so quickly and makes this herbicide so valuable, apart from the issues that we're seeing with resistance, okay, triazine resistance, okay? And so here's, well, you should have that, and here's the pathway, and I'll just show you what, I have some notes here too. This is basically the atrazine. This is what corn does. There are basically 3 pathways that corn uses, okay? Dealkylation, okay, the alkyl group, okay, the NH2 here, okay. So you see this is atrazine. Look at what happens. You got dealkylation, one step, and then a second dealkylation, and this is what the enzyme, the P450 enzyme, is thought to be carrying this out, okay. This process, and you don't have this in your notes, but you should just add it in as I mentioned this, is that this step here is carried out both in leaves and roots of plants. Corn does it, okay? And this process, okay, this step happens in most plants. It's not just corn that does this. So soybean will do it. Some of the oats, you saw, most species can do this step, but it's a relatively minor step, okay, in the breakdown, okay? It takes time, and it's incomplete. That's why soybean does have some tolerance to atrazine. It's not killed right away, but with time, it's just not able to detoxify it fast enough because this is the only pathway that most plants use. Whereas corn is not just going to use this pathway, it's going to use this second pathway, hydrolysis, okay, which occurs in plant roots, and corn does this. Okay, this is an important step, and this Dumboa enzyme, okay, and what you're basically doing is there's dehalogenation. The chlorine, okay, is removed, and a hydroxyl group is added, OH group. Okay, this step is an important step. It's carried out in roots, and corn does this. This is an important way that when you apply, because atrazine can be applied PPI pre and post. When you apply atrazine to the soil, this is how corn breaks it down, the main pathway, because this is carried out in the roots. Okay, so this hydrolysis, dehalogenation, removal of the chlorine, and adding a OH group, this renders it inactive, okay, the atrazine. Corn is pretty well the only plant that can do that, and it's an important step. So it's got that, it's got this, okay, but the soybeans can't do this. So that's why soybeans, that's why when you have atrazine residual activity, which is in the soil, okay, you're going to have soybeans the next year. So you had corn, you rotate to beans next year, you had, residual problems, it's been cool, dry, okay. You've got atrazine in there. You plant your soybean. Soybean does not have this pathway, cannot detoxify. It can do this, but this is applied to the leaves. The atrazine is in the soil. Okay, so that's why you get this problem with residual carryover. Okay, a third step that corn uses is this conjugation. Okay, with glutathione. Okay, which basically adds glycine, cysteine, glycine, these amino acids. It basically makes it into an amino large, okay, bunch of amino acids put together, and this is an important step, and it's carried out in leaves, okay? So corn, again, this is an important pathway. So these two pathways, this is carried out in the roots, this is exclusively in the leaves. Corn does this. It's one of the only species. Why is it corn does it and the others, nobody really knows, okay? Most species will carry out this step, but it's not. As you saw, it's a slow step, and eventually the atrazine gets where? What does atrazine affect? What's the mode of action? The photosynthetic system, remember? Okay? And so the plants that are susceptible can't detoxify it fast enough. They have this step, but this is so slow that by the time you notice 6 hours, they don't, a couple of oats, 2%, 3%, the plant's dead. It goes into the site of action. Corn can do it quick. But however, if you apply too much atrazine, even your corn will be affected. That's why we say too much, because they can't detoxify it fast enough. And so although it can break it, but if you overload the system, it can't, and corn will go as well. That's why you've got to be careful with rates.
How quick from when you apply it post or whatever for that pathway there, how long does that say corn is quicker? How long
Oh, that would be it's 6 hours, and you might have missed this step. This is just showing you corn in 6 hours after application. It's basically it's got 4% left, and that's pretty good. So by the time it gets there, the broadleaf wheats too, they're down. But I mean, you got oats, you got foxtails can ham. 17% is not fast enough. Yes.
Is the pathway for resistance in lamb's quarters one of those, or is it separate?
That's a good question. It's actually does affect, as far as we know, it's a change in the binding site, in one of the binding sites for atrazine.
So
it's not a detoxification. Yeah, it's just basically it's not able. The atrazine gets to the site of action. It's not detoxified. It gets to the site of action. Okay, but the lock and key, basically the lock has changed configuration because of mutation. And we'll talk more about that. But that's a great question. And the trizines is one of the first, 1970 was the first trizine resistant, the common groundsel in Washington State. And I'll take you through what they are. But that's a good question. So again, these are the steps, but that's where you have to be really careful. Okay? That's why also if you don't apply enough of a product, okay, this step, I mean, there isn't enough of the product, low rates, sublethal rates. This step can slow, even though it's slow, it will eventually break it down. Yeah, it might be good in beans, but you've got weeds you want to kill. So don't try to stick to this, to the recommended rates, because they take into account this. That's why, again, atrazine, I mean, it's great, you know, it's a pretty valuable program. Okay, we talked about detoxification. Remember, I've been telling you about, here's the total opposite. We've got species, okay, that basically take a product like 2,4-DB, which is butirac, okay, that's, the product itself is non-toxic. This is used in small seeded legumes, bird's foot trefoil, alfalfa, okay, to control a range of, you know, broadleaf weeds, okay. We apply this, 2,4-DB, basically alfalfa, bird's foot trefoil doesn't do a thing. They leave it as this. This is non-herbicidal. 2,4-DB does not affect the plant. The susceptible species, for whatever reasons, a lot of the broadleaves, the mustards, convert it to 2,4-D, which is herbicidal. It's kind of the total, you know, you always thought, well, the plant's going to try to, you would think that selection would, over time, and there's been some of that, would select for those plants, say, hey, you know, don't convert, you're gonna get hammered, okay? So here's what I call toxification of a herbicide. The reason we could use butirac in alfalfa and small seeded legumes is that they do not carry out this beta oxidation. They leave it as it is, okay? And because, you know, what happens to alfalfa if you use 2,4-D? That's going to be, if you're going to see the flats today and you think I've applied 2,4-D, take a look. It's not going to be pretty. I mean, and dicamba does the same thing, but especially, okay, you get the twisting and bending. So I just wanted to give you an example of here, kind of the total opposite. Here we're talking metabolizing, and this is occurring. And another example, just to show you, is a product. Remember in the greenhouse I told you in wheat, I said we got all these great products for broadleaves, but we have nothing that controls the grasses, okay? Remember I told you in the Great Plains, in the Canadian prairies, even when I was in Eastern Canada, in Quebec, we used this product called dicloflop methyl, whole grass, or hold-on is the trade name, okay? Which is used to control things like wild oats, green foxtails, or the foxtails in things like wheat, okay? And wheat is not affected, okay? And the same thing happens here. Dicloflop methyl is a non-toxic product, okay? It's, well, what happens is basically it's non-toxic. Wheat converts it to dicloflop, okay, which is a toxic product, but it converts it pretty quick and right away then moves it and conjugates it, okay? Adds a hydroxyl and this O-glucose. So wheat goes right through the process. For a short time, it does convert it to dicloflop, which is the active. It's almost like the 2,4-DB example. Imagine this is 2,4-DB, non-herbicidally active, converted to herbicidally active, what would normally kill these grasses. But wheat quickly takes this and has a hydroxyl, the OH, and then a glucose to it, okay? And basically renders it non-toxic. Wild oats don't do that. The foxtails don't do that. What they do is they convert it to the herbicidally active part, and then they try to, you know, some of them will try to add this glucose, but you notice it's double arrows? This is basically a two-way thing. They do it, then it goes back to the herbicidally active, and so the process is such that it's not a clean detoxification. And so basically, the fact that wild oats and the green foxtails are alternating between herbicidal, non-herbicidal, okay, and usually it goes the other way, basically renders them susceptible, okay? Had these, I mean, the idea would have been for the plants and wheat too, you know, not to touch it, just like the alfalfa does. Just leave the dicloflop methyl as it is, and they would have been safe. But they don't. But at least wheat has evolved or just been selected where it actually carries out the full step to get it detoxified. Wild oats have not gotten there yet. Again, is selection pressure what happens? We do have resistance. This is one of those, you know, flops. Okay, so it's an ACCase inhibitor. It controls grasses. It's a graminicide, the one where you, like, you know, Cetuximab, you pull away, you know, the nodes and it's all purplish, mushy. That's it. Okay, but I just want to show you that it's a two-way, okay, and it's unstable. And so this instability causes wild oats to be controlled by this and wheat not to be touched, which is pretty neat. So you've seen examples of complete detoxification, in some cases with Butirac, the tolerant species don't touch it. Leave it as is. The susceptibles break it down to a herbicidal or convert it to herbicidal. Here, it is broken to a herbicidal component, but quickly rendered non-toxic, whereas the wild oats are not capable of doing that. So that's kind of 3 different ways, okay, that plants do. Did the chemists, when they first developed these herbs? No, they probably didn't realize. They just saw, oh, wow, this is affecting this, and then they started understanding what's going on, okay? So take-home message then is, why do we have selectivity? If on prelim 3 I ask you a simple question, tell me what you know about selectivity. Why do we have selectivity of certain herbicides? What are some of the main reasons, if not the main reason? What would you say? It's metabolism. But there are these other examples of height differences and penetration and retention on the leaves. Those all play a role. But by far the major reason is this. Okay? Questions? Okay? And lastly, I just want to show you, I put up this slide because, not only do you have to be careful about, mixing herbicides, especially if they're not on a label, okay, for you to legally mix a certain group of herbicides, but you also have to be careful because sometimes you'll run into what we call herbicidal antagonism. And here's just showing you one of these FOPs, as we call it. So antagonism between a selective post-emergent herbicide, fluazifop, which is one that we are familiar with, and bentazone or aciflorifin. This is a diphenyl ether, okay? And so just want to show you when you have what happens to control. When you use this, let's take fluazifop, okay, as an example, or haloxifop, okay? You use it alone, you get 89% control of Johnson grass, okay? You try to mix it with bentazone because bentazone gives you broader coverage. You can get better broadleaf weed control and some grasses, okay? It's a photosynthetic inhibitor, okay? It stays the same, but look at when you add it to aciflorifin. The two of them are antagonized basically each other, and control goes down to 50, totally unacceptable. Most growers, no way you're going to have 54% control of Johnson grass, okay? And look at quizalofop, and it's based on chemistry, okay? So very often, you do get this antagonism. Now, companies will have already tested the products, okay? And so, and so if they say, yes, this can be safely mixed, you can mix fluazifop with, bentazone, okay? They will take a look at that and they will have tested this. And I remember when we used to do testing for the companies, that's one thing we looked for. The companies would ask, make sure, is there any antagonism? We want to increase. The reason they're doing that is they want to increase the broadness of control. If something's, you got a FOP that's basically controlling grasses, you want something that also controls the broadleaf. We'd say, oh, put them together. But sometimes you do that and you get this antagonism because of the chemistry. So the companies would not put a product like that on the label if they knew it. If they knew, oh, we tested this, it's not a good product. We wouldn't want you to mix this. But they also don't want folks just mixing stuff even on their own, even though it's kind of illegal not to do it if it's not on the label. But also just be aware that that can happen. Okay? Yes?
Does that antagonistic activity happen when you mix them in the tank? Or is that after they're sprayed on?
No, it's usually in the tank together. And that the other thing that reminds me, the other thing is to be sure that the formulations are are compatible. You start mixing things that you'll get precipitation or because of differences in pH of the products or just the way they're formulated. But often it's as soon as they're tank mixed, and when they get onto the plant, they already have they've already caused the antagonism is present. So and that's something I used to test even with biocontrol agents. I was using bentazone and this fungal pathogen. One of the things I was looking for, I was hoping I would get synergism. But the other thing I was looking for, geez, is the bentazone going to wipe out my calototricum? So this happens whether you're working in biological control and mixing herbicides. So it's not strictly, and this is the same for fungicides and insecticides, by the way. And particularly, some folks mix, fungicides with fertilizers and herbicides. Be really careful. make sure it's on the label. And that's why I put that there, not to have you quote that, you know, quizalofop and azathioprine, you're down to 31%, but just be aware of what they talk about when they mean, is there antagonism with these products. If they're on the label, usually, and approved for mixing, the companies tested this out. You're likely not to get antagonism, but follow their recommendations as to which one goes in first and how to mix it, because that might cause you some problems. Okay? So, yes, Bob?
pH control egg life. Do herbicides have a reduced life based on the pH?
Oh, they have reduced life and activity. I mean, just activity. And usually they'll tell you what that pH should be. And particularly this relates also to water, hard water, if you've got it. But the kind of, you know, if you're getting stuff, not just the, you know, hard water, magnesium, but pH levels. Generally speaking, the lower the pH, the more acidic, the faster degradation of the herbicide, generally speaking. But there are differences. So you want to be in that you know, neutral. But often that doesn't happen. So yeah, the pH, reading that, being careful, and again, for those of you who are on farms or using water that is high, you know, hard water, you read the instructions about what needs to be done and what kind of adjuvant you need to put in to balance that. Yes, Joe?
How long you talking like days or hours?
In terms of, with the pH issue, usually. Yeah, I mean, it's not hours, but, you know, a day or so. I mean, you don't want stuff sitting in a tank.
And that's why I was asking, it's not fungicide, you might have only 45 minutes if your pH is too high.
Yeah, that's particular. I mean, in some of the herbicides, that might be the case. I mean, the atrazine can be relatively sensitive to pH. And in some cases, yeah, you wouldn't have that much time. I would always tell growers, I mean, even I've seen growers, you know, where they're spraying, they've got their tank, they leave the tank out in the field because they have to go for lunch or there's an emergency, they have to run, they come back. The stuff is just not the same. I mean, it's photodegraded, it's heated up, pH has clearly changed in there, water's evaporated off out of the tank. And they often, that would be one of the questions that they would, you know, even in these competitions is, you know, and, you know, you talk to the grower, well, I sprayed this in the morning, but I was gone for 6 hours, and it was 90 degrees in the sun, and I left the tank out there. Don't, you know, bring it down in shade, leave it in shade. Try to, you know, if it needs to be agitated, but that is an issue. Fungicides are especially sensitive to pH. That's even more so than I think herbicides. But that's a good point. Again, on the label indicated. Any of these real issues, because that's something we test, you know, our industry does test for these products. Okay? So, I'll leave it at that, and I'll move into herbicide resistance, which is, you know, big issue that a lot of you are going to be dealing with. We're all dealing with right now, and I just want to take you through resistance to herbicides, because this is going to be a major issue. It is becoming a major issue, particularly because of our move to pretty well everything transgenic, where 95% of our soybeans, and I'll show you some data on adoption of transgenics, transgenic crops, particularly herbicide-tolerant crops. It's unbelievable. There has been, as I've mentioned a number of times, no other, okay, basically system that's been adopted or technology faster than this. Than transgenic. So clearly there's something about the you know, and we've talked about it, and I'll give you some examples, and this is kind of, yeah, I think we may need to go back to the hoe and steel. But, you know, people have talked about, you know, could you actually get resistance to, you know, as we call it, resistance to steel, not just herbicides? And, you know, the answer is yeah, of course you could. I mean, look at mowing. I mean, we mow, what happens to the you know, the phenotype of, let's say, a ragweed? Well, it starts growing prostrate, you know, just below the blade. You know, you can't get and we see this in turf systems. How low can you get? You're better off just letting the grass grow and choke out those plants than mowing and giving, you know. So we've worked with ragweed along rights of ways and roadways, and the more we cut it down, the more it would still, you know, was able. Whereas in the, you know, old fields, it's nice and tall, no selection pressure to be for the mowing. So they do adapt. It's not just the herbicides, okay. So, herbicide resistance. is a simple example of what we call classical Darwinian evolution. Okay? And I'll talk about it. Evolution is the result of why have we evolved. Okay? Variability in the genotype. Okay? Genetically, these plants are very diverse. They're not like our crops. That, you know, we could have one genotype in a, you know, you go in the Midwest, it might be the same hybrid, in, you know, thousands of acres, hundreds of acres. Okay? Often this has occurred, variability, there's been mutations, okay, that are spurious. They happen, okay, and 1 out of a million, 1 out of 10 million in the replication process, there's an error occurring in replication that might code for a protein or an enzyme that, you know, in the past wasn't there or wasn't in the same form, or a protein that is the binding site. So for atrazine, the D1 protein changes its configuration. I mean, all that can happen, and from recombination. So it's both a trait of the plant, okay, and then natural selection. You select, we do some things, and that's where we can come in. We do some things that actually will speed up the selection process, okay? Herbicides, the actual application of the herbicide does not cause the mutation. This is something that people, you know, don't realize. They say, oh, you applied a herbicide, it became resistant. It's not what's causing the mutation. Generally speaking, the mutation is out there. It's a naturally occurring, well, like I said, there are 10 million lamb's quarters plants in a field. Maybe one of them has this enzyme that is able to break down atrazine, okay, or it's changed its binding so the atrazine can attach to it. The atrazine didn't cause it, but now what's going to allow it to become the predominant, okay, basically genotype is that we keep using atrazine over and over again, killing the susceptibles, and it is able to reproduce. And I'll take you through that. So what are the things that we can do to reduce the incidence? You know, we can't change the variability to genotypes. These are wild. You know, we can't start domesticating all of them, okay? And the response of a plant to a herbicide is a heritable trait. Once it's in there, it's passed on. That's why you've got lamb's quarters resistance. It's going to be passed on in the seed. And you're going to have all of the individuals coming out of a seed that are going to be resistant to atrazine, trizine resistant. So if you've got trizine resistance, even one plant, and you allow that thing to set seed, it's game over. Then all the progeny are going to be, it's passed over genetically. Okay? And actually, that's not a bad case. I'll show you an example. This is just for trizines, it's what we call maternally inherited. You only would find it in the seed, in the mother part of the plant. We have most of our other herbicide resistance is even transferred in the pollen, both the male part and the female, the ovules. That's where we're running into major trouble, and I'll talk about that. So a double-edged sword. There's no question that herbicide-resistant weed populations are a threat, okay, to the important contribution of herbicides. You may disagree, not agree that herbicides are important, but boy, herbicides have changed the way agriculture has been carried out. If it wasn't for herbicides, the kind of gains in yield and stuff that we have seen, the ability of 1% of the population to feed 99% in this country, or 5%, would not be possible. I mean, I'm not saying, you know, you can agree, but there's at a cost, environmental, others, but no question about their efficacy in general. I mean, I think all of you should take that home, that these are very effective products, and that's why you get that kind of adoption. Most growers are very conservative, they're risk averse, they're not going to get into something if they not going to get a benefit out of it. So recognize that. So the fact that, you know, herbicides are widely used is because they work. Okay? Now we've run into issues, and that's the double-edged sword. Okay? Herbicide-resistant crops, and this is the key, okay? Because we've run into some issues using these non-selective herbicides like Roundup or glyphosate and Liberty, and that's just some examples. I'm not promoting these companies or anything, but just if herbicide-resistant crops, if used appropriately, they really hold the promise. That means to reduce the impact of herbicides, okay, on the environment, get us away maybe from herbicide resistance issues, but I'm afraid to tell you that my personal opinion is that they are not being used wisely. And they're being used widely, and it's the same mode of action here, It's a different mode of action here, but it's, not over many years of application and so forth, we are now starting to see some major resistance. Okay? But the technology is incredible. I mean, and I'll show you the adoption rate. You just have to see the adoption rate, and most of you are on farm. You know what I'm talking about. Why every 99%, 95% of all soybean crop in this country, and about 50% of corn is now in what we call Roundup Ready or transgenics, because it makes control. But this is a big issue, and we're going to run into that, and I'll show you. Okay? So let me just get some terminology straight. Okay? Because you've used some of these. You probably have heard some of these. Like, for example, what's the difference between tolerance and resistance? I mean, I think we all know susceptibility. If something is susceptible, a species, plant injury caused by a herbicide, the inherent plant response to a herbicide. You're susceptible. You get killed. You don't make it. What's tolerant? Okay? The ability of a plant to remain uninjured at the herbicide doses normally used. Okay? The inherent lack of plant response to a herbicide. You're basically tolerant. Okay? You're able to handle it. Corn is tolerant of atrazine. Okay? Resistance. And these two are often confused. Okay? This is very important. The change in plant population from a population that is initially predominantly susceptible, okay, to one that is, okay, to a population that is predominantly tolerant. Loss of effectiveness of a herbicide, okay, due to the evolution of resistance of weed. Good example is lamb squatters. For many years before atrazine, we had, when atrazine was initially used, we had lamb squatters were often susceptible. They were getting killed all the time. Okay? All of a sudden, the population, through selection and the overuse of atrazine, we had a population that was initially susceptible become tolerant. And I'll show you how do you become tolerant. It's at a population level. Okay? What we will call a resistant biotype. Okay? But it's at a population level. Something that was killed in the past, lamb squatters would be hammered, now are not any longer. It has nothing to do with them inherently being tolerant. They were not initially tolerant. Okay? I.e., at some point they were susceptible. They were not from the outset tolerant. Okay? And all of a sudden now they become resistant. That's not the case. These were susceptible, and all of a sudden, over a number of years, they are now, the herbicide is not effective anymore. Okay? That's where you see what we refer to as resistant development in a population, or resistant biotype, or susceptible biotype. Okay? That's not tolerant. Tolerance is inherent. It was from the outset. Do you get the difference? Okay? Naturally occurring. Okay, two other terms, very important. Okay? Something has cross-resistance. If they talk, and this is true for any pesticide. So for those of you in IPM or in plant path, fungicides, the same thing. We talk about cross-resistance. That's resistance that develops to two or more herbicides. You could include in there fungicides, insecticides, okay, if you're working in those areas, with the same mode of action. Okay? So an example would be, I have a weed that's cross-resistant to pendimethalin, prowl, and treflan, trifluralin. They're two different herbicides, but the same mode of action. Okay? So that's bad, but it's not as bad as this. When you run into issues of multiple resistance, I mean, this is still bad, don't get me wrong, but it's at least one mode of action. When you start getting resistance, you get weeds that have developed resistance to two or more herbicides with different chemical families and chemical modes of action. So now you have something that's resistant to atrazine, that's resistant, which is a photosynthetic inhibitor, that's resistant to treflan, okay? That's a mitotic inhibitor, that's resistant to 2,4-D, that's an auxin mimic. You run into trouble. And we have examples of, and I think I have it in your notes, of green foxtail and rigid ryegrass that are, that some weed biotypes are resistant to 9 different modes of action. 9. Australia, Josh, Australia is but they have to be concerned because that's where we have a lot of issues with rigid ryegrass in particular, just because of the way the agriculture being focused on wheat and cereals. But I'll show you some numbers on resistance issues. Okay? So when we have multiple resistance, you have some serious issues. Boy, the wheat has been able to adapt to all these. This is not an easy thing to do, but it tells you, and it depends on basically the genetics of the species as to why some species are able to do that. Yes, John?
So that's incredible. I thought you were about to say, like, maybe there were an example with 3 that were,
No, 9's the most. In Canada, there's some green foxtail that's up to, I think, 6 or 7 modes of action.
So my question is about the rate at which different species become, as a population, become resistant to a herbicide. So because there's sort of herbicide resistance develops over time in weed populations, I kind of get the impression that it develops a lot faster in some species and a lot slower in others.
Yeah.
And obviously that's due to selection pressure, like how much or how intensively herbicides are being applied. But is there other factors determining the rate?
Right.
Because that must have occurred very quickly for 9 to by now,
Right.
9 chemical modes of action to be resistant to it.
We're going to cover that. I'm going to take you through the steps of what are the factors that affect the speed, which species, what are some characteristics that predispose some species to do it. I'll give you right off the bat annuals, much faster development of resistance than perennials. Because why? They produce seed. Genetically diverse, different, and they just, and so you get this mix. The genetics are such that sooner or later you're going to get a mixing in, okay, of variability. So yeah, I'll take you through it and give you some examples. But let's give me. I just want to give you a brief history here. First reported cases of resistance, just so you're aware of it. Okay, to insecticides. 1908, first case of resistance. Okay, 1940 for pathogens. Okay, 1970 for weeds. Okay, now. We can argue that entomology, as I've said, plant pathology, much older fields, well-established. Really, wheat science came to the forefront with the advent of 2,4-D. Very often, as I said, wheat scientists were in an agronomy department. You were an agronomist. You weren't necessarily a wheat scientist. The agronomist knew all that. But you had very specifically plant path, just like we have at Cornell. We have a plant pathology department. We have an entomology department. You do not have a wheat science department because they were generally thrown in there. So this is not surprising. Older fields and some of the projects. This is resistance not just to insecticides, to synthetics, because again, many of the synthetic pesticides were developed in the 40s, but just resistance to even some of the inorganics. Okay, but look at what happened by the late 1980s. Remember, the pesticides really got going. Synthetic pesticides, 40s, 50s, 60s, the glory years, 50s and 60s, and then 70s started going down. 500 insect species resistant, 150 pathogens, both bacteria and fungi, and by 1980s, 50 weed species. Okay? Take a look at presently. I just pulled this, and I'll give you the website. Great website for future reference. Okay? Presently, as of December 1st, yesterday when I pulled this off, 2008, this is worldwide, 183 wheat species, of which 110 are dicots, 73 monocots, resistant to herbicides. In 15 different not all 183 have 15 modes of action, but of those 183, there are 15 different modes of action. I think we've got like 20 or 17 modes of action, depending on which system you use. Okay, but of those, there are actually 311 herbicide-resistant weed biotypes. What we mean by this is you might have, say, a ragweed that's resistant to, this is not the actual case, well, actually, to atrazine, okay? That's an atrazine-resistant biotype, okay? But here we'll just say ragweed once, counted here, but it's also cross-resistant to simazine, okay? So we've got a simazine, which is another trizine herbicide, and so it would be counted here both twice. So that's why this number is much higher. But this is the truth. I mean, of these 311, 183 are actual individual species. But, ragweed and things like, for anybody from the Midwest, there's this water hemp, which is an amaranthus. Amaranthus rudis is a real bad, glyphosate resistant. It's resistant to 3 or 4 different herbicides, it would have been counted in here maybe 5 times. Okay? So this gives you an indication of the individual biota. But if you want to know how many species of all the weeds we know, there are about 183. Okay? This is the website that you should go to for anything. You can look at herbicide by country. You can do it by chemical name. If you want to know, I want to know in Washington State, what are the herbicide-resistant weeds that we know? Okay? You can do it by mode of action. You can do it by weed. You can do it by state. You can do it by country. Mode of action, I want to know all the ACCAs, weeds that are resistant to ACCAs or to this herbicide. That'll give it to you. www.weedscience.com. And it's updated, and we've submitted stuff. I have some stuff I submitted for somebody. So it's for weed scientists and maintained by weed scientists. As we develop, we see some new results, it's put up there. So it's up to date. And that's why I keep changing the numbers here. Okay? Yes?
Is it more current than the Herbicide Resistance Action Committee?
It's HRAC and these guys are similar. They're, you'll see when you visit the site, you can go to both. But that's very similar. But here's this. I pulled all these graphs you're going to see, and I apologize. I didn't get them done in color. Some of the distribution maps that you will see, you don't have to memorize. It was more for you. They're in color, and you can download them as JPEGs off that website if you want and put them in that presentation. And they're, Ian Heap loves for people to use it for their presentations. If you're down the road, you want to present this, it's not a problem. It's not, copyright. They're happy for you to take. That's where I pulled this off. Okay? But look at the development of number of resistant biotypes. Okay? This does not mean resistant species. This is the biotypes. You could have ragweed that's resistant. We have 3 biotypes resistant to 3 different herbicides. Okay? This should be 311. Look at that. 1980s, how it just continued. I mean, it's just first. Remember, herbicides came out. Look at what low, and then in the 1980s, and I'll show you what happened in the 80s. What happened in the 80s? What do you think just got this thing to shoot up? Anybody have any idea of what kind of herbicides came out in the early to mid 80s? Sorry? Glyphosate was 71. ALS is what else? The other group that's, if I ask you, give me the 2 modes of action that have shown the quickest development of resistance. ACCAs and ALS inhibitors, and I'll show you the data. That's where this has come in. And now it seems to have flattened out, but now we're getting the glyphosate resistance popping. It's contributing. Because these things, that's why people started looking at, we're getting resistance issues here. Okay? Distribution of herbicide-resistant biotypes, U.S. and Canada, Australia, 41 or more. Okay? But actually, it's U.S. It's actually, because there's only one country in here. Okay? Canada, Australia, 31 to 40. I mean, you could argue, but the darker, that's obviously based on how developed the agriculture is, availability of the herbicides, but this is where the issues are. Western Europe, Canada, North America, basically, Australia, Tasmania, isn't there? So, and it's increasing, and we're starting to see countries like Argentina and Brazil that are now mass producing, corn and beans, and you guys all know that that's where a lot of our seed comes from, because they have double rotations. Like this, many of the companies grow the seed there, over the winter and then ship it to us ready for spring planting, different hybrids, just because they're able to do that. Okay, again, this is, off Ian Heap's site, but just to give you a feel for that. This might be a little outdated, but I just wanted to give you a sense of, modes of action, MOA, okay, just very general, number of commercial herbicides available. Because the next slide, I'm going to show you the resistance development in these different categories, these different modes of action, and you'll see it's disproportionate. These 2 groups, although they're 30 and 10, are have just shot up. They have by far the greatest development of herbicide resistance. Okay, and we'll talk about just quick. Why do you think that we've seen probably the quickest development of resistance within these 2 modes of action? What is it about them? We've talked a little bit about them. And what is it about them in terms of how the herbicides, what do? They affect what? One, this very specific site. These are very targeted. You use small amounts. So if you get a mutation occurring in that binding site of the weed, just by random mutation, and it changes that conformation. The herbicide can't bind, i.e. it's developed resistance. And then you keep using the same herbicide, that individual will keep going. So take a look at this. This is, again, sorry, it's not in color. It'll be in color if you pull it off. But at least this is up to 2010. It's number of resistant biotypes by mode of action. Okay? Look at these. What's this stuff right here? Okay? Trizines. Okay? It's gone up there too. Okay? ACCase inhibitors. Okay? But remember, it's one thing you should do, and I didn't get a chance to do this, is match up the number of herbicides in that mode of action with the number of resistant cases, and you'll realize that, okay, I think for the ALS inhibitors we have 30 or something, but they have close to 80 or 90 of the herbicide biotype resistance cases. Okay? So these by far, the three of them, the trizines seem to have leveled off. Okay? And look at this, it's kind of low-key. 1970, remember I told you the trizine started? Okay, going, and then these other products, the reason they're low is that they weren't developed till the 1980s. Okay? So this thing, as soon as, 2-3 years after ACCase inhibitors were introduced into the market, that's the FOPs and DIMs. Boom, resistance within 3 years of use. Okay? And the same for ALS inhibitors. Okay? Sulfuron, halosulfuron, all that basic group. Okay? And also, the glycines, what's that, the glycine? What do you think, what herbicide is that, or at least? What's got glycine in the name? Glyphosate and phospho. Look at that. And this matches perfectly with the introduction of Roundup Ready crops. Okay? It was not a problem. Introduced in 1971 by Monsanto. Been used, important herbicide, and then transgenics. And if you match up the adoption rate, number of acreages being sprayed with Roundup, perfect match. Okay? And then you have things like the, paraquat. I mean, you do have it, but it's been used for a long time. The synthetic auxins we're starting to get, but they've been, they were back here, 1942. We're starting to get some, okay? But by the number of synthetic auxins relative to the resistance cases is relatively low, and these products have been used for 60 years, not 3 years, okay? So again, wouldn't ask you, draw these for me, but it's nice information to kind of be aware of, okay? And here's a chart just showing you how quickly, okay, when a number of herbicides, some are actually modes of action, some are specific herbicides. 2,4-D, the actual introduction was 1948, although it's, in the mid-40s. First resistance case, 1990. Took 42 years of use. This is a widely used herbicide. This wasn't something you just put on the side. This was used. Remember we took 42 years. Look at this, ALS inhibitors. Introduced 1982 in general. By 86, 4 years later, we had first resistance cases. And then it just skyrocketed. ACCase inhibitors, 5 years. Very specific, single target site mode of action is its very specific enzyme. Yeah, they're good. They're very narrow. They're very selective. But you run the risk of, if overused, in this case overused, and just basically the weeds figured it out. By random mutation. There was one individual in that population that just had the right conformation, and then by constantly using the same herbicide, we selected them out. The susceptibles died. They became the greater part of the population. Do you guys know what actually, for some of you working in the field, I think I might have mentioned this, when does a grower, generally, even an expert, figure out that they have resistance in a population, a weed population in the field? What percentage of the field has to have the resistant biotypes? Or basically, what is the population of the resistant biotype relative to the susceptible? Okay, so if the whole field is 100, we have lamb squatters, 100% of lamb squatter. What percentage of that lamb squatter has to be resistant for us to actually start really recognizing, whoa, we've got a problem? 30%. I mean, give or take. But it's just to give you an idea. It's not 1%. It's not 50 or 100. It's 30. At 30%, we start going, man, there's something going on here. This is not me. My nozzle getting clogged up. I missed, I missed a row, or it rained. There's an issue going on. Okay? This is glyphosate. 1974, again, after 21 years, but I'll tell you that since 95 to 2008, it's just gone up because of widespread use. Okay? No matter what, it's like anything. You use the same thing too much, it's not gonna, you're going to lose that tool, and that's what I'm afraid we're going to lose. Okay? An excellent product. Again, this is to give you an background. Paraquat took about 10 years for resistance development. Okay? But it's not as widespread as we see it in these other species. Top 10 worldwide. What are some of the species? I mean, Josh had the question, what are some of these species that are, okay? This is the world's worst weeds. There's Home has this book that tells you the worst weeds, just in terms of competition and stuff. But this, in terms of herbicide-resistant weeds, this rigid ryegrass, which is we've got populations in California. Okay, it's mostly in Australia. It's a real issue. South Africa, I believe. And all of this, you can actually see some of this in HEAP. But the ones that we have here, these are here: wild oat, red root pigweed, common lamb's quarters, green, foxtail, goosegrass, kochia. It's in the, you might not know kochia. It's in the lamb's quarters family, Chenopodiaceae. It's here, but it's not as common as in the Midwest and the West. Okay, horseweed, smooth pigweed. Okay, what do you notice about these guys in terms of life habit? All annuals. Okay, so that's one of the things, because they sexually reproduce. Okay, so the question that you have to ask yourself is, I'm likely to get resistance development faster if I'm in an annual cropping system where I'm tilling, okay, than in say in an orchard type situation where I've got permanent. So again, not going to ask you give me number 8 species that's resistant, but just understand. Okay. So one of the questions that I have for you is that, and we used to think this was good, when I taught this course even 10 years ago, I remember I was always proud to say, well, weed scientists, we're lucky because weeds have developed resistance at a slower pace than insects and plant pathogens. And I was really, because you saw the numbers, 150, and we were at 50 by the mid-'80s when I was a grad student, or an undergrad, I should say, but late 80s. I was always saying, wow, look at the difference. Okay, yeah, herbicides came out in the 40s, but insecticides too, and so forth. So I was saying, and everybody, all the weed scientists were pointing to, what were some reasons you think we used to basically justify or say why it is, and some of them are still true today, why has herbicide resistance, was it, at least initially, slower to develop in weeds than, say, insects and pathogens? What would be some things that you would, if I were to ask you that, what would you have thought without reading your notes? Because it's in your notes. Yes, Megan?
2,4-D wasn't very specific. It would disrupt a lot of things that are going on.
Okay. Let's think about the organisms. That's true. Let's think about the organisms as well. What are some differences between weeds and...
Weeds are cold.
So the sedentary, and how would that impact?
Like population becomes isolated, so you can't spread toxins.
Okay, so that might be, okay.
Insects and bacteria and fungi reproduce a lot faster than weeds.
Exactly. I mean, these things can go 2-3 generations, and they're fast, and so you can imagine how fast that's going to happen.
A weed doesn't move, so it's easier to control the dosage than insect.
Right.
So you don't get a half dose.
Okay. Good. Let's think about. There's another important aspect about the weeds that's generally different, and has to do with annuals. And what do annuals do? What they produce seeds, and what happens to the seeds? You guys have learned this. Dormancy.
So you get ones that are like 10 years old that aren't resistant to still growing.
Right. So you get a dilution of the resistance because you have susceptibles that maybe 10 years ago in your cornfield, the lamb's quarters were not resistant, but they can last 10 years in the ground. They're going to pop up now, and what would be nice would be if they would outcompete or be have greater competitive ability than your resistant phytophthalates, which is exactly what was shown for triazine resistance. And all the weed scientists said, at least that's one good thing, they're resistant. But the resistant guys had to pay something. They had to give something up to be resistant. They gave up competitive ability. And we thought, wow, this is cool. As soon as we started seeing the other resistance cases for the other modes of action, turned out not to be true, that this was very specific to the triazines. So ALS inhibitors, species that are resistant, weed species, they're just as competitive as the susceptibles. ACCase resistant weeds are just as competitive, if not more, than the susceptibles, so we lost that. But you're right. that the seed pool, because of dormancy, would dilute the effect. You'd have susceptibles coming up. Okay? So here are some of the examples. You have them in your list. Okay? Weeds normally require full growing season to reproduce. You know, 1 generation per year. We talked about that. That, you know, insects, pathogens. I mean, look at, you know, pathogen. I mean, blight. My God, you know, in a week you could be from not seeing any symptoms to wiping out your tomatoes and potatoes and you name it. So very different in that. Weeds do not move about as freely as do insects. Again, you guys got that. Crop rotations, which usually result in the use of different avoid or delayed development of herbicides. Okay, you could say that's not as true sometimes for insects and pathogens. I mean, yeah, you could rotate and a crop is not as susceptible to a fungus, but this is really true for weeds, much more so. Cultivation or other tillage tends to delay or prevent resistant, you know, that's not necessarily going to affect fungicides, resistance, or insecticides, although it can, but it's not as directly as it does for weeds. Maintenance of a reservoir, we talked about that. The seed bank, okay, a susceptible population because of these dormant seeds. So we thought, oh yeah, these are all factors that would have definitely slowed it down, okay? Resistant weeds can be less competitive, okay? And that's what I just said. Yeah, it turned out to be true for the S-triazines, but not at the time. Remember, this, when we developed this, this was the triazines were where we had seen resistance. We hadn't seen it in some of the others. So we thought, oh, cool, they're all going to be this way. You know, they're resistant, but they're going to maybe be outcompeted by the susceptible. So let's keep the pool of like a refugia, okay? And low persistence, in general, of most herbicides in soil, in general, okay? So they're not going to, you know, that turned out to be not the case. So this is still true, but as you've seen, the way we've been using our herbicides is, you know, is basically we've overcome a lot of these advantages. And that's what I wanted to kind of talk about. So getting back to Josh's question is, so what? What are the factors that affect how fast a species might develop resistance or a population? And there are a number of them that include both a selective pressure, and this is something we can change. What can we do? Okay? Control efficacy, basically effective kill. Okay? Our herbicides are extremely effective. We get 99% of the population wiped out, if not 100% of the population. Okay? So we wipe out those susceptibles really fast. Okay? And in some cases, we have residual activity of some of these herbicides. Okay? So I'm not saying that we should accept less than perfect kill, but that might be one of the things we need to be thinking about. Do we have to have totally clean fields? Should we allow some species that might be, you know, might escape the control? And the argument from industry is that, and folks that say, no, you want the total kill. You don't want like an antibiotic where you're giving small doses to kind of prep you up and you become tolerant. So there's debate as to, you know, what is the best strategy? Is it basically to kill everything, so make sure there's nothing left, or does that encourage, you know, selectivity, resistance? Because the resistant biotype is then going to be, you know, becoming a greater proportion of the population much faster than if you had others, less susceptible species in there. So that's something to think about. We clearly use the same herbicide over with the same mode of action. This is where we're running into with glyphosate. Roundup Ready corn, Roundup Ready soybean. I mean, how could you not get development of resistance? I mean, that is just a no-no. And again, herbicides with single mode of action, we've seen it in the ACCase inhibitors. So these are some things that we'll, you know, factors that are selective pressures that we can modify. Okay? There are some things that are inherent in the plants themselves that there isn't much we could do about it. Okay? I.e., what's the genetic makeup of the particular weed or weed population that you're dealing with? You know, how polymorphic, how many different genetically, how is it able to adapt to situations? How does the resistance inherited? Is it both in the male and the female part of the reproductive structure, or is it just in the female, the maternal part of it? So S-triazines are maternally inherited, the resistance. But for all the other herbicide resistance, you can have them both in the pollen and in the ovule, the male and female. So pollen, that's why there was controversy with transgenic crops, with particularly, say, in the canola, with wild relatives, wild mustard, because the pollen could be transferred and could fertilize a closer related mustard that's and pass on, hybridize and pass on the resistance gene to wild mustard. And then you'd have a problem. Now, Roundup doesn't kill your wild mustard. That should have done it in the past. Yes?
Does resistance develop more quickly in dioecious plants? I don't mean resistance, but as population.
Generally, that's true. That's true, but there are not that many species that are dioecious. But I would say yes because of the ability and combinations of having this mutant in the population. Okay? And we've talked about, I'll continue with relative fitness and the fact that you're an annual weed, okay? And if you're abundant in the population, okay? Let me just see if I'll just leave off with this. I've talked about this, okay? And I'll get back to the other graph. But this is how you get resistance development. So say this is your population, you spray a herbicide. You've got one individual that's a mutant. For whatever reason, DNA replication didn't work out like the rest, and it is now resistant to whatever herbicide. It is not killed. Initially, the population is susceptible, okay? This is a resistant plant. You spray it. Everything else dies. It reproduces, okay? What happens next time you spray the same herbicide, same mode of action? Instead of having one individual, you now have 2 or 3, those seeds, okay? And by the time you spray again, 3rd, 4th year, 5th year, over 30, 50, 60% of the population is the resistant biotype. You've wiped out the susceptibles, okay? And now you're in trouble. Now you've got to start. What else am I going to do? So next class, we'll go through why did resistance development happen in the different modes of action, slower or faster, and what can be done about it. That's going to be, and are transgenics the answer? Okay? So I'll leave it at